So today it's my pleasure to welcome Professor Shelley Halpain. She's a professor of neurobiology at the University of California, San Diego. And she's also has an appointment in the Sanford Consortium for Regenerative Medicine. She's currently in Tokyo, or not Tokyo, in Japan, in Kyoto on sabbatical. And, so we're 13-hour time difference. It's not quite 6 a.m. here, and it's not quite 7 p.m. there. Shelley, could you kind of give some background on you, how you got interested in neuroscience, and then these polymers of proteins, the cytoskeleton that regulates the development of neural circuits and their structural plasticity throughout life? And then we'll go from there.
Okay. Well, first of all, Mark, it's a real pleasure to be on your podcast. So thank you very much for the honor of being here. And I appreciate it very much. So, start back at the beginning. That was several decades ago. I was an undergraduate student at UC Irvine in California. And at the time, when I first started out as an undergraduate, I thought I would become an environmental scientist. And environmental issues are still something that are very close to my heart. But somewhere along the line, I got convinced that neurobiology was a really fascinating topic. And I was, as part of our required biology curriculum for the major at UC Irvine, we had to take a course called psychobiology. And that's kind of an old-fashioned term now. I think everyone uses the term neuroscience instead. But my course was led by a couple of pioneers in neurobiology at the time. A fellow named Gary Lynch was one of them, and I decided to try to do some undergraduate research in his laboratory, and he very kindly invited me to join the lab as an undergrad, and that's where I got hooked. Instead of deciding to go to medical school, I decided that research was my cup of tea, and I thought that I would pursue a PhD. So I went off to the Rockefeller University to do my PhD thesis work. I worked with a wonderful man named Bruce McEwen, who passed away, sadly, way too early, a couple of years ago. And Bruce's lab was one of the leading labs that discovered the impact of steroid hormones on the nervous system. It was thought at the time, before he started doing his pioneering studies, that hormones, particularly steroid hormones like estrogen and cortisol, would work just in the peripheral tissues. And he discovered that there's receptors for these steroids in the brain. And that was a really fascinating idea. So I did my PhD work in Bruce's lab. And then I decided to learn a little more biochemistry type of approaches to studying how the brain works. And I was fortunate enough to get accepted for a postdoc position in the laboratory of the late Paul Greengard. And Paul was really instrumental in providing a huge body of evidence over many decades of the importance of protein phosphorylation as a way. It's a biochemical modification that regulates the activity of proteins. And he was very interested in particular in how phosphorylation impacted the signaling between neurons, and wrote the textbook literally about protein phosphorylation in the nervous system. So I learned a lot from that laboratory about different ways to approach how to study the brain and its connectivity and how neurons signal to one another. And it was in the course of that work that I, sort of by accident, stumbled across a really interesting observation that a very large protein that is very prominent in the brain, very highly expressed in the brain, a protein called MAP2, or microtubule associated protein 2. I found that MAP2 was one of the major targets for regulation by the major excitatory neurotransmitter glutamate. And so I was fascinated, and it turns out that this protein MAP2 is very well known these days protein for studying the nervous system and its structure. And when I was discovering that MAP2's phosphorylation state is regulated in response to neural activity, that was a very exciting discovery to me. And with Paul's blessing, I was able to sort of take that project with me when I left Paul's lab and started up my own independent laboratory as an assistant professor at the University of Virginia. So that was sort of my first real job. And I built upon my previous work studying MAP2 and its regulation through phosphorylation. And that really launched my interest in the neuronal cytoskeleton.
And launch your interest in the world's most important ion, calcium.
Yes, indeed.
Right.
Yes.
And
that is, in fact, where I started to stumble across your papers as I was reading about signals that would mediate the response to glutamate. I started to learning about the central importance of calcium as a signal.
And through major part through regulating phosphorylation of proteins. I remember, so I knew Paul, and when I was at the University of Kentucky, we kind of shifted to studying things that are relevant to Alzheimer's disease a lot. And so and I was on a study section at NIA that reviewed program project grants, which is, a handful of investigators typically at one institution come together and develop kind of a overarching problem they're going to address and then use their different expertise. But we site visited Paul at Rockefeller University. This was before he got the Nobel Prize, but he was pretty much already had the Nobel Prize from a standpoint but anyway, it was really interesting. He was so nervous during the site visit, that was the first time I really encountered him. He was very clear. He was really concerned about every detail of how things went and so on. Yeah. Okay, so you mentioned glutamate receptors. Glutamate's the most prominent neurotransmitter in the brain. Approximately 90% of the neurons deploy glutamate as their neurotransmitter. So it's released from a presynaptic terminal. It binds to receptors. Then there's calcium influx. Then what happens to the cytoskeleton? And I know that's kind of an open, we could spend 2 hours talking about that, but just kind of in general, how does calcium affect microtubules and actin microfilaments, the two kind of main polymer systems?
Right. That's a great question. As you're implying, it's a big question because the cytoskeleton is a really vast network of consisting of many proteins that have to work in coordinated fashion. And indeed, there are many regulatory molecules that regulate aspects of the cytoskeleton, including the microtubules and the actin filaments and the neurofilaments. My expertise is in the area of the microtubules and the actin filaments. But it's important to realize that calcium it works mainly through regulating many different enzymes that bind calcium, and the molecules have a particular shape to them. When calcium will bind to their regulatory sites on the molecule, that changes the molecule's configuration, changes its conformation. And so the molecule now adopts a new shape, and that allows it to adopt a new function. So there are molecules that are enzymes that will be activated when calcium binds to them. And some of those enzymes, many of those enzymes, are calcium-dependent protein kinases. And what a kinase does is it transfers a phosphate group onto a target molecule. And that's the process that we referred to earlier as phosphorylation. And so phosphorylation then, it in turn changes the conformation of the target molecule, and that either turns it on or turns it off, either ramps up its activity or dials down its activity. So there are, for example, cytoskeletal proteins, including MAP2, whose phosphorylation, when the phosphorylation is increased for that molecule, then that alters how that microtubule associated protein, for example, interacts with the microtubule. And so there are protein phosphorylation events on microtubule associated proteins, what I refer to as MAPs, that will cause the MAP to be less able to bind to the microtubule polymer that they regulate. And they kind of fall off. So normally, if you think of the microtubules being decorated with a MAP, then phosphorylation can cause that molecule to lose its binding to the microtubule. And the microtubule then is less regulated by that particular molecule. So in the case of MAP2, what we discovered, which was quite surprising, which people hadn't really paid as much attention to as protein kinase activity, is that we found that there was a dephosphorylation event that affected this MAP called MAP2. And so what happened when calcium was entering the neuron in response to glutamate, then that was actually switching on a different type of enzyme called a protein phosphatase. And protein phosphatases will remove phosphate groups from their target molecules. And in the case of MAP2, that results in MAP2 being less phosphorylated and being more likely to bind to microtubules, which in the case of MAP2, the binding of this MAP to the microtubule should stabilize the microtubule. So we know now, and we kind of take a back step and think about the whole picture of things. There are waves and cascades of responses to a signaling event that allows calcium into the neuron. That calcium event can activate, maybe even sequentially, maybe first protein kinases and then protein phosphatases, and that causes maybe a loosening of the cytoskeleton and then a restabilization in a new configuration. So that's sort of the types of ideas that work from the Greengard lab, my lab, and many, dozens of labs throughout the world have really been, trying to untangle how the cytoskeleton is regulated by these cascades of molecules that impinge on their activity.
And this is important not only after the nervous system forms, but even as neurons differentiate and they grow an axon and multiple dendrites, and then at some point they'll encounter another potential target neuron, or an axon from another neuron will counter dendrites of them. And so back when I was doing my postdoc, it wasn't too much before that there was evidence that neurotransmitters play important roles in the wiring of the circuits during development, because it had been known they act at synapses once synapses are formed, but it was really unknown and not well studied at all that they even act before that. So could you talk about, so as an axon extends or a dendrite, the tip of it, there's a structure called a growth cone. Can you talk about what a growth cone is and how these microtubules and microfilaments are arranged in the growth cone?
Yeah, absolutely. So, as you're saying, Mark, not only is the cytoskeleton important in the mature nervous system for sort of regulating the configuration of the wiring diagram, but during development, it's very, very important when the brain is wiring itself up. Then neurons are often born in parts of the developing nervous system that are far away from their eventual targets. And so the long projecting part of the neuron that we call the axon has to sometimes traverse many millimeters of space, and even longer in the case of neurons that go from your motor cortex down to the bottom of your spinal cord, or from your spinal cord down to your little toe. Those axons have to travel a long distance. And like any vehicle trying to navigate a new city, like you do in Kyoto, you have to have a map. You have to sort of have guidance to get where you're going. And axons are have a specialized tip at the very end of this long protrusion. That specialized tip, as you're referring to, is called the growth cone, and it can be kind of cone shaped, but in textbooks and in certain stages of the growth cone's life, they're more shaped like a hand. They sort of have this splayed out region that you might think of as sort of the palm or base of your hand. And then they have these long little finger-like things that are referred to as filopodia, which are basically finger pods. And that growth cone is filled with cytoskeleton. And the cytoskeleton has to be arranged in a very particular manner in order to help guide and direct the growth cone. And the growth cone's job is to sort of be the sniffer dog of the environment as the axon is growing out. There are receptors all along the growth cone that are sniffing out the molecules in its environment that it encounters, either when it touches other cells or when it encounters soluble molecules that have been released by cells in its surrounding. And those cells, those molecules then cause a signal transduction event in the growth cone, which will redirect the cytoskeleton inside the growth cone to direct a turn of the growth cone, either toward an attractive signal or repelled away from a repellent signal. So that's how growth cones actually are the sort of the steering mechanism for the axons as they're going out to try to reach their ultimate targets in the developing brain and spinal cord.
Yeah. So these filopodia, then they can extend and retract and kind of probe the environment, and there may be a molecule there that they bind to and kind of then move in that direction. So the growth cone can kind of steer depending on the molecular cues it's encountering in its environment. And then also the axon, the growth cone of the axon can release neurotransmitter.
Right.
And we think that that's actually playing a role. So essentially my postdoc work, the take-home messages was that glutamate can affect the outgrowth of dendrites. It turns out that the receptors, so this is, you describe this polarity of the neuron where you have this long there's a cell body that's round, you have this long axon that's projecting, relatively long distances toward its target, and then you have these shorter dendrites, and these dendrites are also tipped by growth cones, but the receptors for glutamate are almost exclusively, or certainly predominantly, in the dendrites. They're not in the axon. So you have a growth cone releasing glutamate, and then if it encounters a dendrite that has glutamate receptors and binds to the receptor, and then a channel opens, which you'll be talking about a lot. It's called the NMDA receptors. It's essentially a calcium channel. So the calcium goes in the dendrite, and then essentially I found that the glutamate release from the axon can ultimately kind of stabilize the interaction between the axon and dendrite, and apparently promote further changes that can lead to the establishment of a synapse.
Exactly. Yeah, there's a lot of motility in the developing brain in particular. And those axons, those growth cones are very dynamic morphologically. These filopodia will extend and retract, and the growth cone kind of will collapse if it's being inhibited to grow out, and then it'll expand when it's got permissive signals around it. And this directs the process of how the nervous system wires itself up. And I know that AI is on the cusp of everybody's thought processes these days, but so far we still don't have computers, thank goodness, that wire themselves up. So the brain can do this, and it's a remarkable process. And the motility of the connected neurons doesn't even stop once the nervous system has matured and formed. Even the synapses themselves have this little bit of motility to them. So
I think you and I,
that's why cytoskeleton is so exciting, because the cytoskeleton is what directs that motility.
I think you and I, well, I guess I don't know this for sure, but some of your early work was in cultured hippocampal neurons, right?
Right. In fact, my present work, a lot of it is still in
yeah, actually you had some, and we'll get to this. You can tell us about two really exciting papers that you know, I wasn't aware of these findings, and you have some really fantastic pictures of growth cones of cultured neurons, and then using antibodies to different proteins that are involved in regulation of the growth cones. But the value of cultured neurons for these kinds of studies is pretty obvious. You can directly look under the microscope and watch these filopodia moving, do kind of time-lapse imaging or that kind of thing. You can actually visualize the cytoskeletal proteins, even in with certain methods in living cells. So maybe go ahead and talk about, you had a paper recently on called INF2. Is that right?
Okay. Yeah. So
that was a new protein to me. I wasn't aware of it, and I think it was in that paper that, if my memory is serving me, you know, I don't know who did the work, you know, the postdoc or graduate students or students that did the work, but really beautiful images of these and how they change when there's changes in calcium influx and other changes, right?
Okay, so the INF2 story is not about development, though. This is the story with the mature neurons that's related, not development. So we could talk about the development story, which is the navigator story, if you want to talk about that, and the growth, or we could shift to talking about what happens when there's pathological signals, which is that INF2 story. So
okay, let's start with the navigators then, I guess.
Okay, yeah. Yeah, that's where I thought you were going with this, was our navigator story.
All right.
Which is, it kind of got us into something quite new. So we started working a few years ago on this set of proteins that really had very little known about them. At the time we started working on Navigator 1, there's a family in mammals, there's three navigator genes, and they're just called Neuron Navigator 1, Neuron Navigator 2, Neuron Navigator 3. But it turns out these proteins are not specific to neurons. They actually are found in many, many different tissues at different levels. We happen to be working on Navigator 1, which is more enriched in the nervous system than in other tissues. But it definitely is present elsewhere too. And you could count on one hand the number of papers that have been published about navigators at that time. And there was very little known about them, but they were described as being associated with the tips of microtubules, specifically at the growing polymerizing end of microtubules. So I don't know, maybe for your audience's benefit, we'll just back up a little bit and explain microtubules. Yeah. So microtubules are polymers that are built from subunits called tubulin, and tubulin has to assemble to build this linear polymer. And it's got its name microtubule. They're very tiny in diameter. They're less than 25 nanometers in diameter. And they are literally tubes. They are hollow down the center. So as they assemble into this cylindrical manner and form these little narrow tubes that serve as the superhighways in the nervous system. So those long axons that we were talking about have linear bundles of microtubules that help support their structure. But very importantly, it also helps transport. It's really the roadways to transport cargo from the cell body, from the trophic center of your cell, the cell body where the nucleus is and where most of the protein synthesis takes place and where vesicles and neurotransmitter vesicles are synthesized, all that has to get down to the very tip, the very end of the distant axon. And microtubules form the tracks for that transport. But those polymers of microtubules are polarized. The one end is different than the other end, and the one end that's more fast growing inside the living cell is called the plus end. It turns out about 2 decades ago, maybe even a little less, there was a discovery made that there are groups of microtubule associated proteins that only or very selectively bind just the growing end of the microtubule. So they're recognizing something structurally that's different about the end. We call these types of proteins plus tip proteins because they're bound to the plus end of the microtubule tip, and they can form clusters of molecules. So it turns out that the navigators had been identified as one of the many dozens of plus tip molecules that bind selectively to the plus end of microtubules through a core complex. And so we were trying to study these, and we were staining with antibodies. We were staining young developing neurons in the culture dish with an antibody to navigator. And we observed, as had been reported, that the navigators seemed to be associated with the plus tips of microtubules. But we also found that the majority of the navigator inside the growth cone was actually in these clusters of, sort of these dense clusters that were highly associated with a subset of actin filaments. Yeah. So actin is the other major cytoskeletal polymer that's really important in developing neurons. And these are even narrower filaments that are formed from twisted helices that are assembled from actin subunits that form these actin filaments. And for some reason, nobody had actually reported before that navigators were associating with actin in this way. And so we just got curious. We wanted to figure out what was navigator doing in this cluster of actin, what's special about these actin filament structures that are not in the filopodia of the growth cone, but they're actually kind of more out here in the palm, more like in the knuckle region of the hand. If you want to use the hand analogy, it's where they're most highly concentrated. And to make a very long story short, what my graduate student, Gina Powers, had discovered after many years of hard work is that those clusters of actin filaments where the navigators are highly enriched, those are sites of endocytosis. That is sites where the growth cone is taking up some of these external molecules in their environment. We think that that navigator, what our paper showed is that navigator has a role in actually responding to, either sensing and or responding to the soluble molecules in the growth cone's environment that serve as signals to tell the growth cone to keep extending or to turn one direction or another. Or to send signals back to the cell body to tell the neuron whether it's on the right track, is it doing the right thing? So we proposed for the first time.
I guess.
Sorry, go ahead.
You can introduce growth factors, which we haven't mentioned at all, right? And there's this evidence that, okay, so you have a growing growth cone and it has receptors for a growth factor, and then it encounters the growth factor. And my understanding is at least some of these growth factors can be kind of internalized in vesicles, right? And then move back towards the cell body. So is that one of the things that's taken up by this micropinocytosis?
Yes, this is exactly what we found is that, as you mentioned, there's some of the signaling molecules that I referred to before that you and I both referred to as guiding the growth cone. Some of these are classes of molecules called neurotrophic factors or neurotrophins. And as one of the many examples of neurotrophins, we asked whether a neurotrophin that we know to be important for these particular types of neurons to respond to as they're growing out, we asked whether that was being taken up by the growth cones and whether Navigator 1 that we were studying is actually regulating the uptake of this neurotrophin. and that is indeed what we found. And the uptake, so the way signal molecules work, as you mentioned before, is that the soluble molecule in the extracellular space binds to a receptor. In the case of neurotrophic factors and other types of guidance molecules, those receptors bound to their ligand get internalized. And there's different ways that cells internalize different molecules. Collectively, internalization is typically referred to as endocytosis, and there's subcategories of them. And there's the type of endocytosis that we discovered that Navigator is involved with is a particular type of what's called bulk endocytosis. What that means is that it's not particularly picky about only looking for one signal. It'll just internalize, engulf, kind of gulp in everything that's fluid in that immediate vicinity, including neurotrophins. So that's what our study had showed, is that this macropinocytosis method of gulping in signals is what Navigator is working on.
And which neurotrophin? BDNF?
Yes, BDNF, brain-derived neurotrophin.
So as you know, so BDNF is easily the most studied neurotrophin in the brain, and it plays critical roles in learning and memory, and the cellular, the synaptic correlates of learning and memory, like what's called long-term potentiation. So are you, or do you plan on looking at the roles of the navigators in synaptic plasticity and learning and memory?
Yeah, I think that's a great question that you're asking. Yes, as you're saying, BDNF has been implicated by many different studies as being important in learned long-term memory and converting short-term memories into long-term memories. And there's evidence that there's evidence from invertebrates that these types of signals at synapses are in fact regulated by Navigator molecules.
Okay.
We don't know if they're not regulated by BDNF per se, but that is a very logical set of experiments to do that we're looking forward to trying. But yes, there's already some implication in synaptic, synaptic activity for the navigators that come from some Drosophila studies.
And so these navigator proteins, they have a kind of interesting structure or characteristics of their amino acid sequence, right? And in reading your paper, there was a lot of focus on this, that their AAA ATPase activity. Is that important for these roles that you're seeing in growth cones of navigator?
If only we knew. That is a great question. The literature up to the time when we were doing studies had all really focused on the cytoskeletal interaction piece of navigator function. The AAA plus activity had never really been, to my knowledge, has not ever been addressed in the scientific literature. And when we started, after we published our paper implicating navigators' function in this signal uptake activity, that's when we wanted to sort of take a step back and look overall at the navigators. Let's look at the gene family. Let's really consider their DNA sequence, their protein sequence, and their evolutionary history for clues as to what they might actually, how is it that navigators are directing the internalization of signals. Scientists like to think in very reductionist ways about structure function. So how is it that the navigator's structure might, control its function? And as we looked and assembled information about, and this is work that I did in collaboration with Robert Hevner at UCSD, we really took a deep dive into the gene and protein sequence and discovered that the most highly conserved region of the molecule is this AAA domain near the C terminus that really hasn't received much experimental attention. So I think that's a very important set of questions we need to ask, is whether this AAA domain is indeed directing its navigator's activities, and if so, how? How is that happening? It's quite a fascinating and big mystery for the navigator field right now.
Because those domains of proteins, so they essentially they use ATP to change the structure of the protein in a way, but to also affect the conformation of proteins they're interacting with. Is that?
Yeah, I mean, we're getting into an area that I am not an expert in at all. This whole AAA domain thing, I think, is fascinating, but I am going to be very candid and say I am not an expert in this. So my read of the literature is that AAA domains can be found in many proteins of many different structures and functions. But the thing that they all do is that they, as you say, they harness the power of ATP. They harness that chemical energy in ATP by hydrolyzing ATP to do work of making large conformational changes in proteins or protein complexes. And so trying to figure out who navigator's partners are, what is it that the AAA domain is doing for navigator and whoever it's interacting with, that's going to be, I think, to me, that's the crux of understanding the navigators better.
Yeah. So we did this one collaboration, and Ted Dawson had this project. I don't know if you know of him. He's at Hopkins. He does a lot of work related to Parkinson's, but they identified, him and his wife, Alina, identified a protein called they named Thorase. And it's one of these AAA proteins, and we helped them, did some electrophysiology and some other things, but kind of we had a cell paper on this. Kind of the bottom line is that this Thorase, through its activity requiring this AAA domain, promotes the internalization of a particular type of glutamate receptor called the AMPA receptor. And so they found that the Thorase knockout mice, you knock this out, and these animals are prone to developing seizures because they're like not turning off the glutamate signal the way. Have you looked at, do you know if the navigators affect the AMPA receptors and affect sensitivity of the cell to glutamate?
That's a great question. We haven't looked, and I don't know if anyone else has looked. But I should mention that I think one of the things that got me even more intrigued about trying to understand the navigator function is that very recently, just before we published our, I think it was right about the time we published our paper about navigator function, but before we published our sort of overall review, our comprehensive review article, a paper came out implicating for the first time one of the navigators in a human neurodevelopmental disorder. And there was a patient identified that had a biallelic mutation in navigator 2, in the navigator 2 gene. It's in protein coding sequence. And one of the mutations in one of the alleles actually falls right in the AAA domain. And the other allele is in another domain that's sort of important and in a coil region that is also in the C-terminal portion of the molecule. So that, again, is really implicating that navigators, number one, play an important role in neural development. This patient has anatomical abnormalities in the cerebellum and in the corpus callosum, is subject to seizures, and has neural developmental delays. And so I think that this is kind of the first clue that, we're kind of on the right track, thinking about navigators functionally in something that's mediating the wiring up of the nervous system in early development.
Wow, that's really exciting. That's a whole, wow, you can jump on that. That's a whole nother NIH grant. Okay, so talk about this process you discovered, called actinification, and the role of INF2. So this is a recent paper in Nature Communications. that you had?
Yeah, we're very excited about this project also. This is kind of at the other end of the lifespan spectrum. We've been studying the impact of overexciting glutamate receptors for a number of years, and we kind of came across more than a decade ago now this really interesting phenomenon in the culture dish, where if you hyperactivate the NMDA receptors, even for just a few minutes, it's not like they're like being bombarded for hours, but just a few minutes of excess activation of NMDA receptors, we discovered induces a really massive reorganization of the actin cytoskeleton. So in mature neurons, normally actin filaments are most highly concentrated in these postsynaptic structures known as dendritic spines. So dendritic spines are just loaded with actin filaments, but the adjacent dendrite shaft is actually, has some actin, but it's mostly fairly devoid of actin relative to the spines. What we observe with the NMDA receptor hyperactivation is that the actin filaments in the spines really extensively depolymerizes, but at the same time, actin polymerizes along the dendrite shaft into really long, very stable actin filaments. The actin filaments in the dendritic spines are actually quite labile. They turn over very rapidly, on the order of, turning over with a half time of 15 to 20 seconds. These filaments in dendrite shaft that build up in response to this this stressful injury condition, are really, really stable. And yet, when the stress goes away, if we remove the NMDA or we remove the stress, those filaments will spontaneously disassemble and reassemble into dendritic spines and take on, as far as we can tell so far, normal configuration of the synapses. But what we got, I really didn't, like I mentioned, we kind of stumbled across this many years ago, and we didn't publish about it because I was reluctant to publish about it unless we, number one, knew what its purpose was, and number two, most importantly, to know whether it actually had a real world counterpart. I mean, does this actually happen in vivo? Now, trying to understand what's happening at the subcellular level in an in vivo model of cell stress, which is basically what this excess NMDA receptor activity represents. It's excess cell stress. It's actually a type of stress that will be induced by hypoxia, ischemia, sometimes seizures, activity, traumatic brain injuries. These are the types of acute injury conditions which will cause excess NMDA receptor activation in vivo and in human patients. But until I actually could find a model system in which we could ask whether this actinification, that's our word for the buildup of actin in the dendrite shaft, which normally doesn't have actin, it becomes actinified. Yeah. I really was kind of like on the fence about what we could really do with that. But fortunately, I got paired up eventually with a postdoc who was a former postdoc at UCSD, Andy Shih, who had his own lab. He's now at Seattle Children's. And he developed this really beautiful model that's perfect for a cell biologist like me, a model of ischemic stroke. And these are like micro strokes. These are strokes that you can induce, that not me, but Andy can induce by blocking a single arteriole in the somatosensory cortex of a mouse. And so he uses a laser that's directed with a 2-photon beam into causing basically a little blockage in a single penetrating arteriole, and causes a little stroke. And what's nice about this model, these mini strokes, is that the infarct is very small and has very sharp borders. Now, the advantage of that from my perspective as a cell biologist is that we know exactly where to look at the subcellular level for changes, for the consequences of that stroke, of that lack of oxygen and glucose supply, right? That's what a stroke is. And what might be happening to respond to this condition. So we were able to actually use this Andy's model and find evidence for this actinification actually occurring within just a few hours after the stroke is induced, at the time where we know excess NMDA receptor activity is happening. And as we were gathering this evidence from this nice in vivo model, we also began to sort of dawn on us from our work, seeing that this actinification is reversible. We began to understand that it's not part of the early stages of cell death that happens after a stroke. This is actually a pro-survival response of the neurons. The neurons actually are deliberately reorganizing their actin cytoskeleton in this manner to protect themselves from the injury. And it is a pro-survival response that if you block it, the injuries get worse, the strokes get worse, the cells are less likely to recover. And the exciting part is that if you encourage this actinification to happen, you make it more likely to happen, that's neuroprotective. So we're excited about this project because we think it's like a real handle on how you might harness the neuron's intrinsic resilience pathways to try to help prevent damage in these acute injury conditions.
And so this apparently protective actinification process, it's initiated by the calcium influx?
Mm-hmm. Yeah, this is an interesting part of the story too. Actinification is definitely requiring calcium influx, but it also requires sodium and chloride influx. It requires the conditions that cause the neuron to swell. So you need calcium and you need the convergence of cell swelling, of water entry, to activate this enzyme that you mentioned that we discovered as the linchpin for this actinification. It's an actin regulatory, or an actin polymerization nucleator called INF2, that stands for inverted formin 2. And our collaborator was one of the early labs that discovered and worked on characterizing INF2 from Henry Higgs. And he, showed that this molecule is normally in a quiescent state. It's normally quiet and inactive in cells, and it needs some signal to come along and activate, change its conformation, open it up so that it's now actively able to promote the polymerization of the actin. So that's sort of the gist of the story that we've discovered, but it's still mysterious. How is it that calcium and swelling are converging to activate INF2? That's a big mystery that we want to try to solve.
And now there are drugs that you've used a lot. We did a little work with this too, that can affect pretty selectively the polymerization state of actin. There's drugs that can cause depolymerization, like cytoskeleton, and others that can, was it jasplikinolide?
Mm-hmm. Yep.
Stabilization. And you use those, right, to kind of manipulate actin and see how that affected the cells, right?
Right. Yeah. We think this actinification, we don't have really strong evidence yet, but we suspect that the depolymerization of the actin from the spines is required to send this to supply actin monomers.
Oh, I see.
To kickstart this actinification. We don't know if that's itself what's activating INF2. There's some evidence in the literature that formins can be activated directly by binding of actin monomers. That's one of the reasons I'm here in Kyoto, is to try to study this a little bit more. But there, at the very minimum, you need a supply of monomer to polymerize the polymer, right? So by manipulating the availability of the actin monomer using drugs like jasplikinolide, we could show that if you prevent the depolymerization of the actin, then you don't get actinification in response to NMDA.
I had a postdoc, Katsutoshi Furukawa. He's in Sendai now at Tohoku. Yeah, Tohoku,
yeah, University.
And actually, So it was interesting. This is kind of a diversion from the topic, but it's kind of an interesting story. So after he did his postdoc, he was finishing up his postdoc when I was at University of Kentucky, and I just got recruited to be lab chief at National Institute on Aging in Baltimore. And so I recruited Katsutoshi as a tenure track investigator, and then eventually he got married and moved back to Japan. But he was when they had the tsunami. So Sendai is the biggest big city in proximity to where the tsunami wave came in. And Katsutoshi had a colleague, and he's a neurologist, and he was actually trained by Akihiko, who was kind of probably should have shared Nobel Prize for a patch clamp discovery, because he was doing pretty much the same thing as the Germans had found at the same time. But anyway, so he had a neurologist friend that was in a 3-story clinic closer, between Sendai and the coast, and they got the tsunami warning, and everybody went to the top floor of the building. And Katsutoshi showed me a video of this. He has a video of his friend took of the wave coming in, and the wave came up to the 2nd floor, but there's all these houses and cars. And then Katsutoshi, he's focused a lot now on Alzheimer's disease and brain aging. And so he did a study and published it, looking at kind of how the elderly people who survived the tsunami, but maybe lost relatives and so on, how their cognition progressed relative to age-matched people. And there's a dramatic decline in their cognitive function, apparently triggered by the trauma, stress of that. But Katsutoshi, when he was in my lab, he did some studies related to actin cytoskeleton and excitotoxicity, which is kind of what you've been talking about. You can get severe overactivation of glutamate receptors, excessive calcium influx. If that calcium stays too high too long, it can actually lead to cell death. But you're studying this kind of early stage where the cell is still able to protect itself, in part at least, through this actinification. But Katsutoshi found if he depolymerizes actin with cytoskeleton, it would reduce calcium influx into the neurons. We were doing a lot of calcium imaging at that time. And then he did some other experiments, had a nice paper with mice where the gene for a protein called gelsolin was knocked out. And gelsolin is a calcium activated actin severing protein. And those animals are more prone to seizures too. So anyway, I thought I'd kind of throw. Do you know whether the INF2 that's involved in this actinification process, if you knock that out in the cells, does it affect the amount of calcium influx or the degree of calcium elevation? Is it somehow feeding back?
That's a great question. We have not studied this yet. That's actually something we're actively working on. So I don't have an answer yet.
Okay.
But based on your colleagues' prior work, I would expect that maybe we will see some action on the calcium influx. I mean, clearly the type of cell stress that we are inducing here with NMDA, either bath applying it in a dish or creating a stroke, that's like a sledgehammer, right? There's a lot of calcium that floods the cell. And as we were talking about earlier, calcium is a, sort of a good guy, a player in trying to regulate the development of the nervous system, the response of synapses in plasticity conditions. But the flip side of calcium, it's like, Dr. Jekyll and Mr. Hyde. I mean, too much calcium is definitely deleterious for the cell.
Yeah.
I'm sorry, I don't know why my camera's
Okay.
acting
Yeah,
up.
yeah, you're getting a little blurry. I don't know what's
I am. Nothing happened here other than maybe it's
Yeah, I was kind of in my
maybe the
mind
internet.
thinking,
Could
is
be
there
the
getting
internet.
fogging of your camera lens or something? Or
I think it's back now.
okay, let's see. There.
Oh, I should mention while you're
I-
thinking, I definitely want to mention one thing that's really important is for me to acknowledge my senior colleague, my longtime collaborator and friend and chief scientific officer who directed these studies on actinification, Barbara Calabrese. So this is really her. She was the driver behind this work.
Now, during our conversation now, I think the listeners and viewers will kind of appreciate the complexity of these proteins that are regulating the structure of the cell. And not only that, but they're not only regulating the structure of the cell, but they're regulating their responsivity to growth factors, maybe to neurotransmitters themselves. And so there's tremendous about to be discovered. You mentioned that in the axons, how long they are, and you've got these microtubules, coursing the length and moving things out, not only synaptic vesicles, but they actually move mitochondria, the energy factory of the cell, out, and maybe mitochondria actually turn over. They don't last forever, so some of them will become dysfunctional, and somehow the cell recognizes that, and then it will either ship them back to the cell body to be degraded, or maybe components recycled. I've always been interested in the energy aspect of growth cones and synapses, but didn't really have a good way to study it. We mentioned calcium, and the late Roger Chen, who was a chemist and kind of a neuroscientist. His brother, who's still alive, is a neuroscientist, Richard. But anyway, Roger, and this was really valuable for my postdoc work, and it just happened, the timing was right. So Roger had developed these fluorescent molecules, and he tweaked them so that the fluorescence changes when calcium binds to them. And he also developed a method where he coupled that calcium sensitive molecule to another, what we call lipophilic molecule, that will allow it to get into cells. So you can just, bottom line is, you can add these calcium probes outside the cell and they'll get in. And so he published the first paper on the first one, called FIRA2. I think it was 1986, and I was doing my postdoc in 86, 87, 88, 89. And we were one of the first labs to use his calcium probe to image calcium in growth cones. And so, but there's no way really yet to image ATP levels, because there's got to be a lot. This is well known in the neuroscience field. So these synapses, because there's all this activation of the glutamate receptors, the calcium influx, the sodium and calcium that move into the cell across the membrane, they have to be quickly removed from the cell to restore the ion gradient across the membrane. And there are energy dependent pumps that pump them out. So kind of my point there is, synapses have a high energy demand. And I'm wondering whether these cytoskeletal changes are somehow affecting the mitochondria movement or association with the kind of under the spine where mitochondria can often exist. So that's and the other thing that kind of prompted me to think about it, there's a lot of ATP dependent molecules involved in moving things. They're called molecular motors. which
Right.
we haven't talked about, and we don't need to go into detail. But there's one called dyneins and kinesins. And so all these processes are energy dependent. I would expect that these cytoskeletal changes you're seeing with the calcium influx, they're happening so rapidly. They're maybe partially energy dependent because you got this AAA protein or domain in there. Have
Right.
you thought about this at all? The kind of interface between local energy availability and the processes you've been studying?
Yeah, absolutely. You're, as usual, Mark, your questions are spot on. I think this is a really fascinating and important question. And, it's been known for quite a long time that, as you mentioned, the brain is very energy hungry. It uses a disproportionate amount of the body's energy. Yeah. And that is largely due. I think there's one area of consensus in this field of brain energy is that the pumps that will maintain and restore ion gradients are major energy consumers, in particular one called the sodium potassium ATPase, which you're well familiar with, I'm sure. So this is an enzyme or a transporter that hydrolyzes ATP to move these ions to rebalance the membrane potential of the neurons. But what the other major consumers of energy are in the nervous system, and how is it that the activity of the brain is so tightly coupled to the usage of energy that the blood flow to areas of the brain is coupled to the activity of the brain. That's what the basis of functional brain imaging is. Functional MRI and PET scans, where we use 2-deoxyglucose. Those are all, reflective of the fact that as brain activity goes up, the demand increases for energy use. And so there's been a number of attempts in recent years to try to develop biosensors to detect ATP, to understand how ATP is coupled to activity and what the major users of ATP are. And so my lab is actually doing some studies now using a new biosensor that we think has some advantages over some of the other biosensors for ATP. And so we're actually able to use this biosensor and look in dendrites and dendritic spines. We're actually not looking in axons or growth cones, sorry to say, but we're focusing our interest in dendritic spines and the postsynaptic activity. And one of the questions we're asking is, I mentioned earlier that in dendritic spines, the actin filaments turn over very rapidly. Yeah. Well, the turnover of actin is itself an ATP consuming process. Actin filaments have an intrinsic ATP hydrolysis activity in them. And so we have begun to ask whether that high activity in just resting basal conditions for dendritic spines, is that a major consumer of ATP? It's too soon to tell to talk about what our results are, but we have a really talented master's student, Lula Katsarillas, who's working with Barbara Calabrese on this project, and we're hoping we can submit a paper later this year on that work.
Wow, that's really cool. And
yeah, we're excited.
I didn't even think of those. So you can just add drugs that either stabilize or depolymerize the actin, see if that affects the ATP consumption. Okay, so I haven't been keeping up. So I retired from the reason I'm doing these podcasts is, so I'm retired from, at Hopkins, I'm just adjunct professor. So I retired from NIA 4 years ago. I've written a couple books, and then I'm doing these podcasts, and so somehow I missed these ATP biosensors.
Yeah, there's just a few, and they're hard to use. It's particularly for really subcellular, work. There's good work in cell bodies and proximal dendrites and stuff, but yeah, it's not that easy to measure ATP. So I think we need new generation of biosensors to really improve things. And one of my collaborators here he's at the University of Tokyo, who modified a published biosensor to make it more amenable to reuse in mammalian cells, has provided his biosensor had a publication so that we can begin experiments on it. So I'm hoping that work will go well.
Okay, well, Shelley, I appreciate you taking the time, out of your sabbatical, and yeah, this was great. I learned a lot. It's getting me excited again about growth cones and the cytoskeleton and spines, and we've learned a lot, and hopefully, people. So these podcasts are intended a lot for obviously people interested in neuroscience, but in particular undergraduates and graduate students, maybe even, that want to kind of see in a conversational setting what the top scientists in the neuroscience field are doing. So thanks a lot.
No, you're very welcome. It was an honor to be here, and thank you for setting the stage. Your work really was asking all the right questions well ahead of the time of your time. So thank you very much for being an inspiration.
Well, I had to kind of keep my foot in the door. So I spend most mornings I'm either writing or reading, or reading your papers or other neuroscience papers and trying to keep up on the field, and then the rest of the day I goof off. So
glad to hear it. Enjoy your time.
All right. Bye.
All right. Bye-bye.